Hello everyone, I am Professor Charu Dogra Rawat from Ramjas College, University of Delhi, and continuing with our series in biotechnology, today I would be discussing about the molecular diagnosis of genetic diseases, specifically about cystic fibrosis and sickle cell anemia. The learning objective for this session would be to understand the molecular diagnosis of cystic fibrosis and sickle cell anemia. As I said, I will be discussing specifically about the RFLP based method today. The reference used for educational purpose is Glick, Pasternak and Patton, Molecular Biotechnology Principles of Application of Recombinant DNA, 4th edition ASM Press. And otherwise the references are mentioned in the slides as they would come. Now, molecular diagnosis is basically the application of molecular biology tools and techniques to identify specific genetic mutations that are responsible for various hereditary diseases. We have been continuing the series in biotechnology, and in biotechnology various tools and techniques are generated as well as applied. So, in this we will specifically talk about the molecular biology tools and techniques that is required or used to identify specific genetic mutations. Initially, the prenatal diagnosis of Mendelian genetic disorders has been limited to conditions for which the biochemical defect was known and expressed in fetal tissues. Now, any kind of genetic disorder which has been inherited by Mendelian inheritance used to be diagnosed by prenatal diagnosis methods, and these methods relied on the biochemical defect or biochemical assays used to be done to identify that what defect is there, but it needs to be expressed, and generally in the fetal tissue, for example the amniotic fluid cells, chorionic villi, blood, skin, liver, etc. But obviously this created or generated a risk for the fetus or the growing fetus. Of more than 2000 known Mendelian disorders, less than 100 are amenable to prenatal diagnosis using the biochemical or morphological markers in fetal tissues. So, this is very important that even if there is some kind of a defect in the tissue, then it has to give some kind of a biochemical assay or produce some kind of a morphological change so that this can be diagnosed. So, as it is mentioned, there are 2000 known Mendelian disorders, but less than 100 could be prenatally diagnosed using these kind of assays. So, there was a need actually to develop more molecular diagnostic techniques which is going to use efficient biotechnological techniques and tools so that the diagnosis can be more efficient. So, the diagnosis started with the DNA based diagnosis methods, and one of the method that we will be discussing today is the RFLP based method. Now, based on the DNA isolation from the tissue is much easier rather than identifying or taking out the tissues for the biochemical assay where it requires a very large amount. So, from very smaller amounts the DNA diagnostic tests can be done. RFLP, if we just brush up a little, the RFLP is restriction fragment length polymorphism that refers to the differences or variations in the DNA sequence at sites recognized by the restriction enzymes. Now, we all know that in the DNA there are sites for different restriction enzymes. Restriction enzymes are the endonucleases that cut the DNA at specific sites. So, these restriction sites are widespread all over the DNA, they are in the sequence, intrinsic in the sequences, and there are differences or variations that occur. Now, in fact, each human being is unique based on these restriction sites that are present, because there are certain markers due to which these sites can differ. So, no two organisms RFLP print is basically the same, just like the fingerprints. And if you remember, and you can look at the other session, we had discussed this RFLPs when we were discussing about the DNA fingerprinting. because in DNA fingerprinting these RFLP markers were used to identify the differences in the two individuals. So, these markers are basically arising because of the differences in the restriction enzymes sites, and therefore, when they cut you get different length fragments. So, such variation results in different sized or length DNA fragments produced by digesting the DNA with the restriction enzyme. So, no two DNA, even the parent's DNA, some of the bands are always different. Some of the fragments are always different because these restriction sites are differentially distributed in the DNA of various organisms. And because of the differences in the site's location, when they are digested by a particular restriction enzyme, they give fragments of different length. So, this restriction fragment length polymorphism is a very important tool to be used for molecular diagnosis of genetic diseases. The SNPs or indels, the SNPs is basically the single nucleotide polymorphism, or insertions and deletions, the indels can create or abolish restriction endonucleases recognition sites, thus affecting quantities and length of DNA fragments resulting from the RE digestion. So, when we talk about that the restriction sites are differently distributed, this is because of these two things and others as well, but these two things such as the single nucleotide polymorphisms. So, single nucleotide polymorphism means that there is a difference in a single nucleotide in various DNA, and that can occur because of the replication errors and mutations. There can be spontaneous mutations, or there can be mutations which is basically triggered by certain mutagens. So, these SNPs arises, or as we know the insertions and the deletions, which is again occurring because of the mutations, and these can be used as the markers so that they will abolish or create. Generally they abolish, but they can also create restriction endonuclease recognition sites, and therefore when we digest the DNA with the restriction enzyme, then we get different numbers or different length of the fragments that are obtained. So, this is a very important tool that is harnessed for the DNA fingerprinting as well as for molecular diagnosis of diseases. So, this is the picture that basically shows that what I am talking about, that if you see that there are two restriction enzyme sites, now because of certain variations you can see that one of the sites could have been removed as well, or because again there are some kind of mutations or some changes that have occurred that have created another site. So, this is the abolishing of a particular site, this is the creating of another site, this is the movement, the location, displacement, it was present here, but now the site has generated here. And then again there can be the other one can be removed, or this is again a kind of a displacement which is a far away from this thing, and this is mentioned here due to the presence of VNTRs. So, what are VNTRs? VNTRs are the variable number of tandem repeats. That is, in between the two restriction sites there would be tandem means one after the another, repeated sequences which are present. Now, each block of repeated sequence can occur variable number of times in different DNAs. So, in some of the DNA there could be 2 tandem repeats, in some there can be 10, and in some there can be 50 or 100. So, obviously the distance of these restriction sites would vary, and therefore when we cut with these two restriction enzymes, then basically the length of this fragment will differ, because in the length of the fragment if there are 4 or 5 tandem repeats it would be smaller, if there are 50 tandem repeats it would be larger. So, when we run it on agarose gel electrophoresis or gel electrophoresis, when we do, we will observe different length fragments on the gels, and therefore you will have a different print for various different DNAs that we are studying. So, these are the basically arising the creation or the abolishing or displacement of different kinds of restriction enzymes or restriction enzyme recognition sites. So, they can occur because of the single nucleotide polymorphisms, they can occur because of the insertions or deletions, and they can occur because of these VNTRs. Then there are other markers such as STRs, which are the short tandem repeats as well. So, these are basically the different kind of markers that give rise to polymorphism in the restriction fragment length. This again explains, in a normal and a diseased manner. So, what I was saying that, for example, in here, due to mutation or certain action of a mutagen or whatever, this particular site has been abolished. So, when this site is abolished and when you cut the DNA with this restriction enzyme and you run it on an agarose gel electrophoresis, so in the agarose gel electrophoresis you can see there will be a different sized fragments, and in these different sized fragments you can see that the two bands appear here in this particular lane, which is for example for a normal DNA, and this is basically for the this diseased DNA. You can see only a single fragment appears. So, there are two fragments for this and there is a single fragment for this, and you probe it with this particular gene, what we are talking about, or this region of DNA when you probe it, and when you southern hybridize it, this is a southern blot. So, we have already discussed in detail about all these techniques. So, when you run the gel electrophoresis and you transfer it to a nitrocellulose membrane and you probe it with this particular region of DNA, you would obtain different size fragments. So, you will obtain two fragments in this lane and a single fragment, large fragment in this lane. So, this way you can see, you can actually observe and diagnose that this is the normal DNA because it gives two bands, and this is probably some change has occurred, and it can be from the a diseased person that the difference in the fragment has occurred. So, this is how the RFLP is basically used, and this is depicting how the restriction fragment length polymorphism can occur. So, two different lengths could be obtained depending upon whether the restriction enzyme recognition site is abolished or it is intact. Now, when we talk about diagnosis, there are two general approaches. One is a direct method diagnosis. In the direct method diagnosis, DNA from the at-risk fetus is directly tested for the presence or absence of the abnormal gene. Now, what do you mean by at-risk fetus? At-risk fetus is identified based on the pedigree. So, if you identify or you look at the family history, and in the family history or in the family DNA analysis, you find that the parents are heterozygotes, then there is a possibility that a particular disorder can be, it depends upon the disorder how it is inherited. If for example, it is a recessive homozygous condition, it is there, or in a heterozygous in a dominant homozygous condition, it is here. So, identifying the inheritance of that particular gene and knowing about the the family history, if we identify that it is the parents are heterozygotes or it is present in the parents, then basically at risk fetus are tested or diagnosed for predicting whether they would be carrying the disease or not. So, in this case, they are directly tested for the presence or absence of the abnormal gene. And another method could be the indirect method diagnosis. We will look at the examples that sometimes that it is not, the gene is not basically showing any polymorphism as far as the restriction fragment length is concerned, but a linked gene or a linked area which lie within approximately like 1000 kilobase pairs to the disease gene, they identify the RFLPs or they show polymorphism. So, again why linked? We again understand about the linkage, that how the linked genes are inherited, that has also been talked about in detail in previous lectures. So, when the linked genes, because the linked genes tend to inherit together, they do not separate during the recombination. The mutations is a different thing, right? But when we are talking about the inheritance, the two genes which are lying very close to each other, they are inherited together. So, if there is no disruption or in creation or abolishment or any change in the diseased gene, but there is a change in a linked gene which is present very near to it, then this diseased gene inheritance would be dependent or would be linked to the nearby area. So again, this will behave in a similar manner. We will look at these method of diagnosis as well. So these are the two molecular methods, the direct method or the indirect method diagnosis that we would be studying. Some of the diseases that can be identified by the direct method of diagnosis are the sickle cell anemia, beta thalassemia and alpha thalassemia, and then there is a deficiency, the trypsin deficiency, DMD, Duchenne muscular dystrophy, cystic fibrosis, congenital adrenal hyperplasia or the fragile X syndrome. We talked about this again when we were doing karyotyping. So, it is a X-linked mental retardation. Now, these diseases in DMD, cystic fibrosis or congenital adrenal hyperplasia or hyperplasia, we need to know what kind of mutation is there in the gene. Then only it can be used for the direct method of diagnosis. If this particular gene we are not sure that what kind of mutation is present, then we cannot identify it by the direct method of diagnosis. So, we will look at that. We'll as I said, we will take up the examples of sickle cell anemia and cystic fibrosis and we'll get to know in detail. But cystic fibrosis can also be identified by the direct method of diagnosis, provided that we need to have a family or a relative that is basically showing the disease and we can identify what kind of mutation is basically causing the disease. The indirect method of diagnosis can be used to diagnose these different diseases. Some of the diseases such as cystic fibrosis or DMD or beta thalassemia, they can be identified if they are just randomly they are occurring and the gene has been cloned. If the gene has been cloned, the disease in which the gene has been cloned, but the mutation is not known, then we can basically use the indirect diagnosis method. And some of the diseases in which the gene has not been cloned as well, for example, Huntington disease or for example, dystrophy, the myotonic dystrophy and muscle atrophy, so spinal muscle atrophy. So, in these diseases where the gene is not identified, then again we can use the linked gene method or the indirect method of diagnosis, the linkage studies. So, in cystic fibrosis, we will take up both the examples where we can do a direct method diagnosis and where we can do an indirect method diagnosis. So, these are the different methods of diagnosis which is done. So, in the direct detection of mutations, what is the procedure? The blood cells or any other tissue cells are basically taken and then the DNA is isolated from them. This DNA is then PCR amplified by using the primers carrying these different tags. So, when this DNA is PCR amplified, then you obtain large number of those fragments of that particular gene. As I said that for direct detection of mutations, the gene or the region where the mutation has occurred need to be known, so that we can use PCR primers to amplify that particular region or gene and then we digest them. So, this gives rise to a restriction fragment length polymorphism, because the restriction enzyme sites would be impaired in these particular DNA. They will be occurring at different points. We talked about that just now, that if it is a normal DNA or if it is a carrier DNA or if it is a diseased DNA, you will get different length fragments because of the abolishment, deletion or change in the location or creation of the restriction enzyme recognition sites. So, you will obtain different fragments. When you run it on the gel and then you identify, for example, in this case you are identifying based on the tags that have been incorporated. So, this is showing you the location of these particular fragments. So, when they are scanned, then they will give different sized fragments, and then based on these we can basically identify whether the mutation is there or the mutation is not there. So, this can diagnose whether that gene is a diseased gene or it is a normal gene. So, I hope you understand this direct detection of mutation, because this polymorphism, the restriction fragment length polymorphism has occurred in the gene or the region that we are studying. So, it is a direct detection method. In the indirect detection of mutations, it is a marker identification. So, first of all we have to identify, as I said, in the indirect detection of mutations, we do not know the mutation. The gene has been cloned, one category was the one in which the gene has been cloned, but we do not know what mutations, because a number of mutations can give rise to that particular disease. So, we do not know what mutation has occurred in that gene, and then we have to, if we have to study the linkage, if we have to do the linkage studies, then we need to identify the marker, that what marker are we going to look at which is present in next in the 1000 base pair, kilo base pair area near that our gene. We need to identify that marker. Obviously, it should be polymorphic in nature and we need to identify that. Then the next step is the family genotyping. So, again in this indirect detection mechanism, we need to have the family. We need to identify more number of people so as to we can ascertain that what is the polymorphic nature of that particular marker, and then what is the manifestation of that particular disease based on these particular polymorphism in the marker that is occurring. So, we need to genotype the family. Genotype is basically knowing their genetic makeup, that what is the genetic makeup pertaining to that particular gene we are talking about. And then we track its inheritance. So, we create a pedigree and we track its inheritance, that that particular gene, how is it inherited, what is the manifestation of disease in the family, in the relatives for that particular gene or for that particular mutation or disease that is there. So, this is the indirect detection of mutations that is occurring. This is again showing just an example that after the identification of marker, the family recruitment is done where we genotype the family members or basically the individuals. As you can see, in this particular case, this is a carrier and this is a non-diseased male, and they have this DNA. This is just an example to explain. So, they have the wild type allele, but this has a particular mutated allele which is present, and that is why it is a carrier, because one is a wild type or a normal and another is a diseased one. Then when they have the progeny, then both the progeny are carriers. Now, when these carriers are basically married to again the normal individuals, then when these carrier again they have the carrier offsprings, and when they marry together, they will have the diseased one. So, this is a typical Mendelian inheritance we are talking about. So, wherever there is a heterozygosity for a particular allele, it becomes a carrier. Where there is a homozygosity for a particular allele, in this case it is normal, and when there is a homozygosity for a particular allele which is mutated, then it becomes basically the diseased one. So, this is a recessive pedigree. So, if you remember, this is a recessive inheritance we are talking about, because whenever there is only one mutated allele, it is a carrier, it is not showing the disease, it is showing the disease in a homozygous recessive condition only. So, the dominant as well as the heterozygote is are basically the carriers, but the homozygous recessive is the diseased one. So, this is basically how the inheritance pattern occur, and this is basically then we need to identify. So, we need to recruit the family, we need to identify the marker, and we need to identify the mutation in the DNA, and then in the diseased state we need to diagnose whether the disease will be manifested or not. So, this is an indirect method of DNA test. Now, direct test can be employed. So, there are certain limitations. Obviously, the indirect test is advantageous, because there we need not know the mutation, and sometimes we need not know even the gene, and we can diagnose a particular disease. But again, both the direct and indirect method has certain pros and certain limitations are there. Now, direct test, the limitation is that it can be employed only if the specific mutation in that family is known. The applicability of the direct method is thus limited to genetic diseases in which the precise molecular defect is known. because we are identifying the polymorphism in that particular gene, then we need to have an idea of the specific mutation that is occurring in the family. Then only we can, diagnose whether it will be there in the or whether it is there or it will be there in the progeny or not. So, this is a limited aspect. In indirect method, one of the requisite is that multiple family members, usually included at least one living affected relative, must be available. So, as I said, in the indirect method, the availability of multiple family members and particularly one living affected relative is very, very important, because even if we do not know the particular gene or the particular mutation that is occurring, we need to know that the disease is running in the family. So, we need to have a living affected relative so that we can isolate the DNA, and then we can basically predict or identify first of all the marker, so that the inheritance of that particular gene can be studied and therefore molecular diagnosis can be done. Then this indirect method is costly and time consuming as well. Then the third limitation is the possibility of genetic recombination in one of the parent's gametes between the disease gene and the linked marker. This is again very important, because when we are talking about the linked markers, we assume that linked marker and the genes are inherited together. So, but there is not even 100% chance that they will be linked. Sometimes there can be genetic recombination occurring between the linked genes as well. So, if there are recombinants, then we cannot use that marker to ascertain the inheritance of the diseased gene. Although the probability is less and it depends upon the genetic distance. Again, you can go back and review the lecture on the linkage analysis. We can find out the distance between two linked genes based upon the number of recombinants that is appearing in the progeny. So, if the distance is less, then the genes tend to be 100% linked. There will be no recombinants but all the parental combinations. But if the genes are far apart, so further apart they are, more are the number of recombinants between them. Now, several different linked RFLPs that map to either side of the defective gene should be used basically. So, if we have large number of linked RFLPs that we are studying, then it is basically more authentic to be used as a diagnostic method and ascertain the defective gene that should be used. So, we continue with the particular diseases and their molecular diagnosis in our next session. Thank you.